Okay, this is the podcast for STEM Biology, Chapter 5, Cell Transport. A lot of this stuff we did cover in class. On your drawing, you should have labeled the polar heads. Each polar head has two non-polar tails that are hydrophobic. This whole thing makes up the phospholipid bilayer. Within the phospholipid bilayer, there are proteins. Some of them extend all the way through. Some of them extend only part way through. And this is the cell membrane. Here's the outside of the cell, and here's the inside of the cell. Now, things can go through this membrane. The cell membrane is responsible for allowing things in and out of the cell. Well, there's two ways that can happen: passive transport and active transport. Some of this we covered in class already. Passive transport is the movement of a substance through the cell membranes without the use of ATP. There's no energy required. There's three main types. First type is diffusion. Diffusion is defined as the random movement of a substance from a high concentration to a low concentration. Diffusion will continuously occur until equilibrium. And that means that concentration is equal inside and outside of the cell. Here's diffusion. Here's a lump of sugar being put into this water. Over time, the water will have an equal amount of sugar dissolved in it. Three terms to know in this section. Solute is a substance that dissolves in water. That's like your sugar and your salt. A solvent is the water, and when you mix those two together, that is your solution. All right, the second type of transport is osmosis. Osmosis is the diffusion of water. So it's the same thing as diffusion, except it's water moving from a high concentration to a low concentration. It is passive transport, so there is no ATP required. So if you look at this diagram, there are 9 water molecules on this side of the membrane. There are only 6 on this membrane, so water will diffuse across the membrane by osmosis to equal it out. Increased water pressure inside the cell is called osmotic pressure. When water is entering a cell, that's going to cause the cell to get a lot of pressure in the vacuoles and on the membrane. When water leaves the cell, it's the opposite effect. So there's actually 3 effects of osmosis: hypotonic, isotonic, and hypertonic. Hypotonic first. Hypotonic is defined as the solute concentration in the environment is lower than what's inside the cell. So this would be an example of a cell that's in not a lot of solute water, like just pure water. If a cell is in that type of environment, water then is going to move into the cell, into the vacuoles. The vacuoles are going to get really big. They're going to push on the cell membrane. And if it's an animal cell, the cell could burst, okay, like a firecracker. Plant cells will not burst, ever, because of that strong polysaccharide cell wall. Isotonic environment, the solute concentration in the environment is equal to that inside the cell. Everything is at equilibrium. Nothing's going to happen. Most cells are this way, and it's basically a peaceful, natural environment for the cell. The third one is hypertonic environment, and this is when the solute concentration in the environment is higher than what's in the cell. So that means this cell is in a very salty environment on the outside of its membrane, or a very sugary environment. And so what will happen? In this situation, water is going to move out. Water always moves from a high concentration inside the cell to outside the cell, in this case. So water will move out, and that causes your cells to shrivel up. All right, so here is a chart, if you want to put this into your notes, that helps you put all these ideas together. For a hypotonic environment, hypo means low. What that means is that outside the cell is low solute, inside the cell is high solute. In reference to that, outside the cell has a lot more water than inside the cell. Water moves from high to low. So water is moving from outside the cell to inside the cell, which means the water will go into the cell and the cell could possibly burst. Isotonic means equal. That means there's equal amounts of solute and water inside and outside the cell. Nothing's going to happen overall. And then a hypertonic environment means high. So that means high solute outside the cell, low solute inside the cell. In reference, high water, there's more water inside the cell than outside the cell, so water is going to leave the cell, and that causes the cell to shrivel up. Okay, you can see this with red blood cells. Here is an isotonic red blood cell. Water is moving in and out. Everything, the cell is basically staying the same size. That's isotonic. If the cell is in a hypotonic environment, water is moving into that red blood cell, and you can see it's swelling up and could burst. It's not, this is a natural one. And the water is moving out in a hypertonic environment, and that causes the cell to shrivel up. Okay? The last type of passive transport that I want to talk about in this podcast is facilitated diffusion. Facilitated diffusion is the transport of specific particles. So we're talking about something specific that has to go through proteins that are in the membrane. We drew those proteins in our diagram. Now we're going to have things going through those proteins to get into the cell. That's facilitated diffusion. Membranes are selectively permeable. In this case, the movement for facilitated diffusion is still high to low. So here's facilitated diffusion. Here are our molecules, whatever it is that's specific. It's going to go through these proteins to get inside the cell, as you can see, and they let them in. When a protein is involved, that is when it's called facilitated diffusion. Otherwise, if it's just going through the regular polar heads and non-polar tails, it's regular diffusion. And that finishes up our podcast.